Okay, hi everybody. Thanks for joining us today in the Genetics and Genomics session. My name's Kevin Holden. I am the head of science at Synthego, and I'm going to be talking to you today about the development of an arrayed whole human genome gene knockout library using an optimized multi-sgRNA CRISPR approach. And just kind of for the agenda today, I'm going to tell you a little bit about what the company that I work as a scientist for, Synthego, does, and also how we've optimized CRISPR approaches for generating gene knockouts, like are used in the library I'm going to tell you about today, and then how we've actually gone around designing and building this arrayed whole human genome knockout library. And then lastly, how you may be able to use this library as a powerful resource in your own research applications. So just kind of, so we're all on the same page, going to be talking a lot about utilizing the CRISPR-Cas9 technology to engineer gene knockouts and how they can actually accelerate your own research and development. And so kind of the old way that people used to do gene knockouts if they're trying to attain a gene knockout was to use a technology called siRNA, or the silencing RNA. And while this was quite effective for some people, it really was not a complete gene knockout. It's more of a gene knockdown. It's very transient in nature, so you're actually not making a permanent genetic change. You're just making kind of a transient gene knockdown. And one of the real big drawbacks of this technology is that you tended to see kind of these off-target effects where essentially these short RNA molecules could affect the activity translation of other genes that were perhaps unintended targets. So it really did kind of make generating gene knockouts not the most effective way. And so there really are some good reasons why you may want to knock a gene out in your own research. These can include trying to discover the function of a protein. It could also be to validate an antibody that you developed against a particular gene, or for validating an assay that you're working in. And then also it could be that you're trying to analyze a particular pathway in a cell, and one way to perturb different nodes in that pathway in order to understand it better is to actually knock out the genes and the associated proteins that are components of that pathway. And something else we'll talk about today is using gene knockouts in this library format for doing functional genomic screening. So this could, it could be that you're working on drug discovery, you're trying to identify a new therapeutic target or validate a therapeutic target. It could be that you're interested in repurposing a drug or small molecule, so screening a compound that's already known to be active in some way to see if it can affect other proteins inside of a cell, or for understanding concepts such as synthetic lethality, and understanding how you can initiate, for example, apoptosis in a cell. And so just so we're all on the same page, just to kind of back out a little bit, we're going to be talking about the CRISPR-Cas9 technology for generating gene knockouts. It's a very powerful molecular biology tool. Essentially, it's utilizing a strand of RNA that is used to program a nuclease, in this particular case, the Cas9 nuclease. It programs that nuclease to a specific location on a genome, and then initiates a double-stranded break. And a powerful tool because once you initiate this double-stranded break, genomes have to repair themselves, so in order for cells to stay alive. And they can do that in one of two ways. On the left, we can see the non-homologous end joining, or NHEJ, repair pathway. This is when you make a double-stranded break in a genome, and essentially the cell repairs itself using a lot of this double-stranded break protein, these double-stranded break protein repair pathways. And these can end up forming what's called an insertion or deletion, or an indel. And typically, if these indels form in a region that's coding for a particular protein, then these can lead to a frame shift, which can lead to a gene knockout. And we're going to mostly talk about that today. Also, this technology, as you may know, can be used to do what's called homology-directed repair, where you supply a donor DNA fragment, typically a single-stranded piece of DNA, as a template in order for you to generate specific single nucleotide variants or specific knock-ins and program really specific changes into the genome. And so there are many different applications for this technology in both disease research and therapeutics. We can see here that you can utilize these reagents, so the Cas9 nuclease and the guide RNA, and you can directly inject those into an embryo of an organism to build a model, so for a mouse or for a zebrafish or a fruit fly. And you can also put them directly into cells. It could be a primary cell, like a T cell, or it could be a stem cell. Maybe you want to develop an organoid. And then they can also be used to generate in vitro models of disease in, for example, cell lines. But they can also be used to do high throughput screening. We're going to talk about that mostly today. And then there's really the big promise of CRISPR from the therapeutic side. So maybe if you identify a target through high throughput screening that you're interested in developing a gene therapy for, these components can be used to do in vivo gene editing, so directly affecting a genetically diseased organ in a person, or these reagents can be used to edit cells that come from a patient, are edited in vitro and then ex vivo transplanted back into the patient. So perhaps a primary T cell or a stem cell of some kind. But just to kind of also give you an overview of what Synthego as a company does, and I'll talk a little bit about why and how we developed this library and the approach that we've taken, really is to provide, the goal of the company is really to provide access to genome engineering tools. And so kind of the first thing we did in order to do that was we developed a new synthesis architecture for making the RNA molecule components. So the long single guide RNA molecules. And really, we were the first company to make a factory that could mass produce these. And they're chemically modified and 100 nucleotides in length. And so really, in order to generate libraries, you really need to have the capabilities to synthesize large amounts of this material. So why do we chemically modify these single guides? Well, this is really a landmark paper that came from the lab of Matthew Porteous of Stanford University, showing that if you chemically modified these single guide RNAs and you use them in this duplex with Cas9 nuclease, you could essentially edit very effectively these primary human cells like T cells or stem cells. And they showed that you could really go from just doing, forming these indels in a low percentage, 2 to 5% of the cells in your population, all the way up to 80 to 90%. So this really was kind of a landmark paper in the CRISPR-Cas9 field. And so what we've done as a company is we took that approach and we figured out a way to mass produce and synthesize these RNA molecules. And so in 2016, we launched a factory that could make these things using a novel synthesis architecture that's controlled entirely by software. And this factory actually is capable of synthesizing over 1.5 million guide RNAs per year, either in tubes or actually in these library plates as well, which I'll talk about today. We can make it all the way up to gram scale as well. So we're actually focusing on building a GMP pipeline so we can essentially support researchers from discovery all the way from the bench into the clinic. And if you're curious to know what this factory we've generated looks like, there's a new documentary film that premiered at South by Southwest this year, 2019, produced by Dan Rather and Adam Bolt. That actually talks a lot about the CRISPR-Cas9 technology, and the Synthego factory is featured in this film. You can see in the window in the top right there, along with your favorite CRISPR friends, Jennifer Doudna, George Church, Feng Zhang. So if you're interested, please check out humannatureFilm.com, and you can see what the Synthego RNA factory is all about. So enough of the movie plugs. Going back to the science, really we work with mostly with this RNP complex format. So it stands for ribonucleoproteins, where you have a purified Cas9 nuclease, you've duplexed it with these chemically modified synthetic single guide RNAs, and this really was kind of state of the art going back about 3 years to 2016 now. And so really is the most effective way to deliver CRISPR into a cell. I'll also be talking today about how other methods that you can deliver the RNA directly into your cells for screening, for example. But using these RNPs, they're much easier to transfect into cells than plasmids. They're transient, so you don't get these off-target rates that you can get with constitutive expression of the CRISPR components in a plasmid. You get very high editing rates, and it's very common to be able to form indels in your genes up to the 90% range. And you can knock in single nucleotide variants quite effectively as well. And really a powerful advantage of using this format is that you don't need to use any antibiotics or selection in your screens or your editing in general. We can see that when we use these RNPs in numerous cell types using methods like nucleofection, so electroporation, that you really can effectively form indels in a wide range of different cell types, ranging from cancer cell lines to suspension cells, primary human T cells, iPS lines, or animal cells. And again, we mostly have focused on synthesizing these single long guide RNAs, the 100 nucleotide approach. We have looked at the older two-part systems, but you can appreciate from this graph here that we've been able to obtain some of these two-part systems from other vendors. And when we compare them, we just really see that the single guides are much more active across a wide range of different targets. This screen was done in HEC293 cells, but essentially as a provider of genome engineering tools, and we're trying to provide the best access to customers, we focused on the single guide format because as you can see, and we found in our own research, and you can find yourself, these really are the most effective way to engineer cells using CRISPR-Cas9. Here we're looking at a 384-well plate, electroporation plate, nucleofection, where we've taken the same guide RNA and we've consistently nucleofected it into 384 different pools of cells. And you can see that they're remarkably consistent. And this is great because it tells us that the process we use for making this material is very consistent as well. So you can start to get very reproducible experiments. This is really important if you're doing a screen. You want to make sure that the material you're using in your library in the screen is very high quality, and it's not going to have a lot of variation between experimental replicates. And so just to kind of back up and talk a little bit about what we've done, we've developed this consistent synthesis method for making these RNA molecules that as you'll see later, are used in this library that we developed. It gives you very reproducible results. And it turns out it's very applicable to, because you get a lot of reproducibility for automation and doing CRISPR at scale and generating large data sets like a screen. In the course of doing this, we've had to develop some bioinformatic tools that can help design and also analyze our CRISPR experiments, and I'll talk a little bit about those before we move on to the library. The first is the Synthego ICE tool, so it stands for Inference of CRISPR Edits. This is a tool that we developed internally for our own use, but is also free for any researcher to use online. There's an online portal for this. And so we have a submission in the bioRxiv. We can read all about the algorithms we developed in order to build this tool, but essentially it's a way of analyzing CRISPR editing using PCR of the region you're trying to edit in your cells or your cell pool, and then getting Sanger sequencing data back, which you can literally get back the next day, and then doing a very quick analysis of that population of cells to determine the editing frequency. And in the bioRxiv submission, you can see there's very good correlation with more time-consuming and expensive methods like next-generation sequencing. So this is really good, a really good proxy for understanding exactly what's happening in your cells with your CRISPR editing. And we made this tool so we could do high-through-put editing analysis. So in this particular example, we're looking at 500 different samples that we've done CRISPR-Cas9 editing in, individual wells. And so you can batch upload large data sets using this tool, which is really great for doing large-scale genomic analysis, which is something you may do in a screen in order to check the quality of the screen. So you can just drag these files in a zip file into this web interface. Again, it's free to use. It's very useful. And the outputs you can see is you get these very nice visualizations that can actually demonstrate the contribution of specific genotypes in a pool of cells. And we give you back an ICE score, which stands for the amount of CRISPR editing you're seeing, and also a knockout score, which is the percentage of those indels that can actually generate a frame shift. So this typically means that they're indels that are non-divisible by 3, so they can actually generate a nonsense frame shift in your coding region. This tool also has knock-in analysis, which is very useful for other applications. But essentially, doing CRISPR and high throughput has allowed us to build better on-target models for understanding how we can predict CRISPR activity. And here we're just looking at several different models that predict CRISPR activity, and then comparing them to what we actually observe when we use the RNP approach. And you can see that the correlations are quite poor. So essentially, this means that many of the guide RNAs that are recommended using these different design tools, if you will, really are kind of under-predicting the activity, the true activity of these guide RNAs. And so, as a company, we thought, can we actually predict these ourselves? This is data we first presented at the Cold Spring Harbor CRISPR-Cas9 conference last year in 2018 that showed that we can actually predict the distribution of the indels that form based on the sequence of the guide that you're using in the localized region. And so this is very powerful because we can actually predict indels before we actually do the experiment. So this allows us to control and understand better what the RNA that we're synthesizing and if it's going to be effective in generating a gene knockout. So doing this in silico prediction was very useful to us. And because we developed these tools for analyzing CRISPR and high throughput, we can get very effective CRISPR editing, we started to think as a company, can we actually automate this part of the CRISPR workflow to build these in vitro models for generating high throughput? And this also got us thinking, like, if we're going to generate these in vitro models of disease and we're going to knock genes out, what's really the best way of doing that? And so, again, we really wanted to try to enable access and standardization of this technology to many scientists. And so we kind of thought, hey, let's build a platform where we can automate CRISPR cell engineering in high throughput. And if you want to do that, it turns out it's actually quite difficult because you have to take into consideration all of these different factors if you're trying to build an automated system for doing CRISPR editing, and how you design your experiments and set them up, how you actually do the transfection, optimize that transfection, and how you manipulate cells, edit them, and isolate your clones. And this is true whether you're doing cell line generation or high throughput screening. And really, this is kind of an overview of all the things we do at Synthego in a software-driven process to do gene editing in our cells, but it breaks down into kind of these 5 categories: designing material, like I mentioned, optimizing transfection, and then doing editing analysis and clonal expansion of cells. But if you're working in a library, that's not so important. So we developed this solution for automating cell lines in high throughput, and we started to think about what's the best way that we can optimize protocols and methods for maximizing gene knockouts and gene knock-ins. So if you want to build a CRISPR high throughput platform, it really you want to start by doing the easiest things first, like a gene knockout, and then focusing on the hard things like a knock-in later. So we really wanted to try to understand how we could optimize editing in virtually any cell line. And this will help us not only figure out how to do CRISPR editing and high throughput for cell line generation, but also for doing CRISPR screenings in virtually any cell line. And so every time we bring a new cell line into Synthego, we run it through an optimization protocol where we essentially test over 200 conditions, and in a very few days, we can really move up the optimal editing efficiency from protocols that are often given by different research groups or companies that make the electroporation device, for example, and we can push this up into regions that makes it very practical and worthwhile doing. So in this case, we moved from 77% to 72%, and that's great because now you don't have to do so you can really rely that what you're generating in your screen or your cell line actually has the edit that you want. And so we've done this for multiple different cell lines now. We've done all of these types of optimizations. But going back to what I was saying, really we wanted to try to figure out if we're going to do CRISPR and high throughput, what's really the best way that we can kind of guarantee that we're always going to get a knockout of a gene. So we're going to start to do gene knockout projects. We maybe want to go into doing some functional screening on our own. So how can we really guarantee that you can always knock a gene out? What's the best way of doing this? And so, again, I'm mostly going to be talking about, and the data I'll show you today is mostly focused on using RNPs, the RNP approach, and nucleofecting them. And then a little bit later, I'll talk about how there are some other methods you can use to get the CRISPR reagents into different types of cells or cell lines. And so really kind of the old way of trying to knock a gene out in the early days is you would use one of those scoring algorithms like I showed you earlier that maybe don't predict so well. And you would actually go through and you'd pick 3 or 4 different guide RNAs and find the PAM sequences and then do some sort of analysis. In this particular case, we're looking at ICE analysis. So this is Sanger sequencing analysis in order to determine which one is forming, which guide is forming indels at the highest rate. So on the left, we can see we've done some editing in these in this MEL C2 C12 cell line of the MyoD1 gene. In the middle, we're done some editing in this PGP1 iPSC line. And on the right, we're trying to edit these primary human T cells. And you can see there's really kind of a wide range of different genotypes, if you will, that are outcomes of picking different guide RNAs in different regions. So again, we're trying to knock a gene out. So we're targeting an early exon in a primary transcript of a gene in order to generate these indels, these non-homologous end joining indels. So we can make a frame shift and then get a subsequent protein knockout. So this seemed like a good approach, but there really are some drawbacks to doing this. And the first being that you often have to screen several guide RNAs to find the best one that works. So we developed a tool previously, like I mentioned, to kind of predict this activity. And it's pretty good at doing that, but not perfect. Sometimes you can get guide RNAs that actually don't perform very well in these early exons. So if that happens, you're just not going to get a very strong knockout phenotype because you just can't form indels in high enough percentages in those early exons. And then also very important is that not all indels can actually cause frame shifts. And below is some ICE analysis where we can see that there are some genotypes in the sample we looked at that form these -3 or -6 indels. And they're essentially, because they're derivatives of 3, which is the same as a codon, they're essentially probably not going to cause a frame shift leading to a protein knockout. That's a problem. So we started thinking, like, we had a hypothesis, can we actually mix some of these guide RNAs together and see if they can work together? And so what we would do is we would go through and at the beginning, we'd pick several of these top scoring guide RNAs based on algorithms like the Dench algorithm I showed you earlier in that chart, and try to generate a fragment deletion that you could use to cause a frame shift. And so we kind of considered just kind of pooling all of these guide RNAs together, maybe picking the top 4 or 5 scoring guide RNAs in a particular region of a gene and seeing if they work together. There were some technical issues with this. The first being that before we developed the ICE tool for analysis, the existing Sanger sequencing analysis tools, like the TIDE tool, really couldn't handle some of these deletions that we were generating by mixing the guides together and getting kind of multiple CRISPR cuts in the same region. So before this came out, we were really kind of just recommending that people use single guides to knock genes out. But what we could see when we mix these together, and here we're actually looking at the analysis from using the ICE tool that we developed and also the TIDE tool, which is kind of the legacy tool for Sanger sequencing analysis. We could see that when we added 2 or 3 of these guide RNAs together, and here we're editing in a HEC293 line, you could kind of get this combinatorial effect that we could actually only see using the ICE tool. And that's because we were mixing these guys together and they were generating these larger fragment deletions, maybe 50 base pairs or greater in this early exon of a gene. And so if you cause this fragment deletion in an early exon, it's definitely going to cause a gene knockout. So it's going to be a problem, and whatever you transcribe after that is essentially going to be nonsense. And then, but again, the ICE tool was only able to identify the larger fragments than the TIDE tool could, but we saw that adding 2 or 3 guides together, we really were able to dramatically increase the activity and kind of the knockout compared to using a single guide. And so that, like I mentioned, that fragment deletion genotype seemed to be really favored strongly. And again, it leads to a strong corresponding knockout phenotype. So we tried this again with several of the cell lines and different locations and different genes. And we can see that when we actually use 3 guide RNAs, over 2, we could get more of a consistent result, so more of a consistent high rate of editing, so more of a consistent pattern of this fragment deletion. And so we looked at some larger data sets, and here we can see on the left the individual guide RNAs, and on the right we've kind of multiplexed these together. And you can see there's a bit of an increase here from about 65%, a little bit over 70% of the total indels that you're seeing in this population. And that's great, so we're getting a bit of a bump here. But several things to consider that are important is, one is that these are actually just indels we're looking at here, and so they may not even cause frame shifts. And the other thing was we weren't completely sure if using the spacing that we were using in this small region, maybe 50 base pairs or so, was this good enough and was this important? And literally we couldn't analyze anything bigger than that. So we went back to the software development team and we're able to develop a tool, an add-on for the ICE tool that allowed us to actually accept 3 different guide RNAs in a region of about 1 kb. And so this allowed us to deconvolute these more complex genotypes from these larger fragment deletions. So we decided to generate a larger data set from a larger experiment where we took over about 100 genes with 500 unique single guide RNAs in them that represented maybe about 1,700 predicted different editing outcomes. And so what we're looking at here is actually the end-to-end spacing and how far apart we space these guide RNAs. And as you can see, we kind of biased ourselves in this experiment because we were considering that the guide RNAs that are closer together would actually work more effectively in a cooperative manner if they were close together. And so the negative numbers here actually mean guide RNAs that are overlapping. And we thought this would be the most effective way of doing this. And so when we first did the first screen, we looked at the first set of guide RNAs that went out to about 80 bases, 80 base pairs in spacing. We could see that there was kind of this wide range of activity of efficiency of indels that we were forming. But interestingly, there was this kind of subpopulation on the right here where we had guides that were spaced very far apart. And we could see that we were always getting high levels of editing efficiency when we spaced these guides really far apart. So we decided to look at the rest of the guides that were spaced really far apart. And we found that when we spaced them even further, we really were kind of pushing to get this high level of efficiency every single time. And we didn't see kind of these wide spectrums of editing scenarios here. And so what we're actually looking at is in the blue here, you can see the single guides when they're kind of close together versus the green ones. And so essentially what's happening here is when you have guides that are very close together, sort of the most active guide RNA will kind of take over. And you'll get most of the editing genotypes from that most active guide RNA. But when you mix these together over a space, you can see that they're actually cooperating together in many different kinds of ways. And so what's actually happening here, like I mentioned, we're mixing several guides together in a localized region. And so they're forming this fragment deletion. We're doing this in an early exon, and this drives a very strong knockout phenotype. And so sometimes you can have some of these guide RNAs actually don't work very effectively on their own, but when you mix them with other guide RNAs that may be some distance away from those, you can see that they act in this kind of cooperative fashion. And what we're looking at here is the difference between mixing these guides together in this kind of multi-guide RNA multiplex format minus the activity of the best single guide. You can appreciate that very few on the left-hand side here actually work better individually than when they're combined with their friends, so to speak. And on the right, you can see in this kind of blow up that you can sometimes get these extreme cases where you have 2 RNAs, guide RNAs that look like they have virtually no activity. But when you mix them with another guide RNA, you can kind of completely recover that poor phenotype. And so what we think sometimes may be happening is that sometimes these RNAs will actually cut inside the cell, and the repair is actually seamless. And so we don't see an indel being formed. We see a cut and a resection essentially back together very quickly. But when you, and you can't individually identify those, but when you mix those with another guide RNA, you can see that you form these dropouts. And another way of looking at this data here is the scatter plot. Is you can see on the top that when you mix these guys together, they work just much more effectively together than they do on their own. We took a subset of this data, just these 60 genes here out of the 100 that were interesting. And on the left, you can see that there's a wide range of indels that form in these 60 different individual guide RNAs, and when you multiplex them together, you can really move up that particular genotype. But what's important though is we're just looking at indels, and if you're looking at the graph on the right, this is the knockout score. So again, this is the percentage of indels that are non-derivative of 3, or maybe they make a larger deletion greater than 20 bases that is strong enough to cause a gene knockout. And you can see that there's a wide range in those same guide RNAs. Most of those guide RNAs cannot form a frame shift, but when you mix them with other guide RNAs, you're much more likely to generate a dropout, which you can see from there. So really mixing the guides together was giving us a very strong knockout phenotype. And we decided to try to understand if we can actually predict how these patterns were occurring. So in the top, in the 3 boxes, you can see the red lines actually are where we actually had spaced the guides together. And the blue lines represent where we actually see the editing. And you can see that those blue lines always fall on one of the red lines. So based on our spacing, we're always getting a dropout and an editing event that's exactly where we think it is. So there's not a whole lot of kind of random cutting that's occurring here, and this really means that there's a mechanism at work here that's actually predictable. And this is really powerful because it means that mixing these guides together, you can actually kind of bias your editing experience if you're trying to form a gene knockout towards getting the highest cutting efficiency that's going to give you a reliable genetic outcome, in this particular case, a gene knockout. And so it turns out that perhaps the directionality and target strand of these guides can be important, and this kind of complicates the design. So what we did as a company is develop an algorithm that actually designs these guide RNAs with kind of the optimal spacing, along with their optimal predicted efficiency, and the least amount of off-target effects. And we've done this essentially for every gene now in the human genome, so we can actually predict and synthesize 3 guide RNAs that we're confident will give a gene knockout phenotype at any particular locus. And so we started thinking, hey, we developed this cool tool that we wanted essentially to utilize to make these in vitro models to form strong gene knockouts. Could we use this tool? Could we use these findings that we discovered to essentially build a better gene knockout library, a good gene knockout strategy where you can make a library of all of these knockouts for the whole human genome? And so we've really tried to utilize this multiplex guide RNA and what we've learned from these experiments into leveraging that into building this arrayed whole human genome CRISPR knockout library, which is, I'll talk about the development of that right now. So just to remind you, again, we're using multiple guide RNAs to drive this fragment deletion genotype that can give us a strong knockout phenotype. And it turns out that, like, if you want to build a library, an arrayed library for generating gene knockouts, it's really important how you actually build these libraries. So we decided to build these in either 96 or 3D4 well plates. Essentially what we're doing is we're mixing 3 of these guide RNAs that are going to work together in an optimized fashion to make that fragment deletion into a single well into each one of these on these library plates. Of course, leaving spaces like we do empty for controls and things like this. And so essentially what we're trying to do in this library is almost guarantee that in every well, you're going to get a gene knockout. So we're really trying to make it so researchers don't have to waste their time and go back and rescreen wells that didn't form the gene knockout. And really doing in this arrayed approach helps you to actually later, if you want to, kind of go back and mix and match these together. Why did we decide to develop an array library? Well, aside from the fact that we're synthesizing these single guide RNAs individually, mixing together, there are a lot of advantages to doing arrayed library screening compared to things like pooled library screening, for example, whereas the pooled library screening is very good for doing large-scale screens. I'll argue that the arrayed library can be as well if you have the right automation set up. But essentially, using the pooled approach, you really are limited by the types of phenotypes that you can identify. There's definitely bias in representation in those libraries, and also the targets that you can hit as well. And more importantly, when you work in that pooled approach, you kind of have to go back at the end and deconvolute using some kind of bioinformatics tool, like exactly what targets that you're hitting. The arrayed library screening, obviously there's no deconvolution. You don't have any representation bias in your screen. And more importantly, you can actually screen virtually any cell type using these modified single guide RNAs, either as an RNP or a different format. And so you can also screen virtually any phenotype. So this allows you to do a lot more than you can do in these pooled screens. There are some drawbacks, of course. It's a little more expensive to do this. You have to buy the reagents, and you do need to have some automation set up as well. But it's a really powerful way of doing doing a library screen of gene knockouts. We decided when we designed this library, to try to be a little bit smarter about how we actually design it. So we looked at other libraries that people were developing out there for using these CRISPR knockouts, and we could see that a lot of them really underrepresented the number of genes in particular protein families, and we were curious why. So we decided to actually go back and use multiple different sources to identify all the genes in a particular protein family or pathway. And so really we're trying to be a little bit smart about this and maximize the number of targets that could be assayed in a particular library, and it's very important. So like I mentioned, we've actually now done bioinformatic knockout design for the whole human genome, and also we have broken these down into different protein functions, protein families. I'll also say that you can actually get these libraries in any kind of custom format. So you can build your own family of genes for a particular library, and that's something that we can offer too. Some of the metrics associated with this library, we were able to actually design this 3 multi-guide approach for over 90% of all the genes of the almost 20,000 genes in the human genome that we're targeting. There are some that are it's very difficult to design for. That's kind of just part of dealing with biology. And there are some where we had to kind of relax some of the rules that we utilize for picking these guides, like the off-target, for example, which may not be so important in a large library screen. And so how do these libraries behave? Here's some examples of genotypic analysis. Here we were knocking out this gene, MRPL12, and you can see here from these genotypes, this is the ICE analysis using Sanger sequencing, and the 3 different guide RNA approach, that we're getting a very high knockout score. So 84% of all the cells in this pool of cells have a genotypic knockout at least. And so we're seeing these large deletions, minus 130, minus 129. That's definitely going to cause a gene knockout when it's in an early exon. And again, here's one where we're seeing 100% gene knockout. This is actually quite common. It may seem unbelievable, but it's very easy sometimes for us to get almost the entire cell population have this fragment deletion genotype. And this also works well. Sometimes I showed earlier, you can only use 2 guides because that's all that's available in that region, but you can also get strong knockouts this way as well. And we can prove this to ourselves by doing things like Western blotting in order to prove that we don't have any protein present in this particular case. We're just looking at 3 different genes here where we knocked out CINP, the COSI, or the CDK9, where we just did some proteomic analysis on the back end using these Westerns to prove that we didn't have any more protein after using the multi-single guide approach to knock these genes out. So how might this library that we developed be useful in your research? Obviously, this technique is very useful to us for generating cell lines or for doing our own screening as well, but it may be useful to you. Like I mentioned, kind of the old way of doing this was using the silencing RNA approach. Again, that comes with its own issues, mostly that it's transient. It's not a real knockout. It's more of a knockdown. So after you apply these into cells, and after some time, the effect goes away, so you may not be seeing essentially the complete picture of what you're trying to look for. And again, using these knockouts, as I mentioned earlier, can be very useful if you're trying to discover the function of a protein or validate an assay or a particular antibody that you developed, or also doing pathway analysis inside of your cell and perturbing those different nodes to understand exactly the role of a particular protein in a pathway you might be studying. And again, using these for functional genomics is kind of really the power of utilizing these gene knockout libraries. And we feel we have a really good approach here for maximizing these gene knockouts in these libraries. So when you do these large-scale screens, you're not going to be wasting your time with having genes that you actually can't knock out in your screen. Essentially knock out all of the genes that you want to target. So how can you deliver these arrayed libraries? Well, these chemically modified guides can actually be delivered in different ways. Like I mentioned, they can be delivered as RNPs directly into cells. That could be a cell line. It could be a primary cell. But you can also deliver these guide RNAs alongside Cas9 mRNA. That works also quite well. Or also in cell lines that are already expressing Cas9, so they have constitutive Cas9 expression or maybe Cas9 expression that you can turn on or off. And again, these can be delivered, all of these formats, RNPs, just guide RNAs, or with the mRNA, using tools like electroporation, nucleofection, which you can do in high throughput. You can get a 96 well or a 3D4 well nucleofector instrument, or they can also be delivered using techniques such as lipofectamine. And so here we're just seeing an example of a small library that we've built where we delivered the RNPs using nucleofection into primary human T cells. So to be able to do a screen in primary cells is actually quite powerful and something that was quite difficult before. And so here we're seeing that we can get these very high knockout scores. Sometimes they're not always high as you'd like, but in general, you're getting a lot of protein knockout here in the small library of 28 genes. And the viability that we're seeing is also very high, particularly compared to when we do a mock transfection. So really not losing a lot in terms of cells when we're combining these guide RNAs together to form these dropouts in these cells. And here's some data that we generated in collaboration with Jonathan Weissman's lab at University of California, San Francisco, UCSF, where they also delivered the RNPs into these primary human dendritic cells using nucleofection. They happen to do next generation sequencing analysis. And what they did is they compared using a single guide on the left of each one of these charts here in green to mixing the guides together, which is on the far right of each of these boxes, or also individually. And you can appreciate, if you look in the box for TNF here, you can see that mixing the three guides together gives you a very strong large deletion. And in the middle one here for the TLR4, we can see that the single guide they were using that was predicted to work quite well using this dense prediction algorithm actually performed quite poorly. But multiplexing these three guides together, they got a very strong genotypic knockout by NGS. So essentially what you're doing with this approach is kind of hedging your bets, and you're essentially making sure that you don't miss any genes that you could potentially be knocking out that maybe didn't work just using a single guide approach. And really getting these guides to work together in a small region is very important. As we've seen that sometimes if you just kind of randomly pick guides across the entire gene, they could be 10 or 20 exons away, and they're not going to work together. So they kind of need to be in this controlled environment. And again, the algorithm we've developed is very useful for that. And so this is the quote from a researcher, a postdoc in Jonathan Weissman's lab. Essentially, it's exactly what we were hoping to do when we developed this library, is to help researchers like Marco here really utilize this multiplex guide RNA approach so they can kind of get the most effective knockout efficiency. And again, this has really helped their work and has improved the sensitivity of their screens by really enhancing their ability to detect some of these weak phenotypes that they could get from knockouts maybe they weren't seeing using their previous single guide approach. You can also deliver these guide RNAs, or these arrayed libraries, into Cas9-expressing lines. It's a little hard to see this data here, but what we've done is we've actually tried to knock out the track locus in this Cas9-expressing HEC293 cell line using the CRISPR Max lipofectamine reagent. So we're using RNPs here. We're using single guides here into the Cas9-expressing line. And so what's really important is that when you use just the guide into a cell line that's expressing the Cas9, you can actually use a lot less of the guide RNA. So you can literally get hundreds of screens out of one of these libraries compared to maybe 4 or 5 screens if you're using the RNP approach. And what you really need to take away from this is that you actually have to optimize this for every Cas9 expressing line that you work with, because you really don't know what the Cas9 expressing level, what the level of that Cas9 protein is in that cell. So in this particular case, we took, just to go back to that slide, we took different ratios of the lipofectamine and the amounts, the concentration of the guide RNA that we're delivering into cells in order to find kind of the optimal range for doing that. And you can see here that when you do these optimization type experiments, you get a wide range of different genotypes, and it's very important to do this every time you work with a new cell line and you're doing a screen, so you can make sure that you can really maximize that gene knockout and find the optimized transfection conditions for your cell line. And you can see how these look also in the ICE tool. So you can put these 3 guide RNAs in and really understand what are the contributing genotypes that you see in these wells to understand that you're getting a strong knockout genotype. And we also have protocols that are available on our website for helping you optimize these if you're working with liquefaction type reagents as well. And so really, there were tons of assays you can do once you've got your screen optimized and up and running. The amount of arrays, the amount of assays you can do with these array libraries is tremendous. So what you could be looking at things like cell survival, doing single cell analysis, and maybe doing some kind of phenotypic readout that's image-based or fax, or actually doing a whole well analysis using some kind of reporter gene or luciferase, for example. That's just kind of wrapping up everything that I've talked about today. We've been able to generate this very effective methodology for knocking genes out. We developed this because it was very important for us as a company to develop these tools so we could enable our cell engineering platform. It's a technology that we trust in order to knock genes out very effectively. And we've now actually kind of shifted this over, kind of tech transferred this into building a library that's an array library for the whole human genome as a knockout. And then, so what you'll actually start to see also is that if you want to knock out individual genes, we'll actually also start to produce these kits that actually focus on individual genes as well. And that'll be a product you'll see soon from the research labs here at Synthego. So thank you for watching. If you have any questions that were about any of the data that was presented during this talk, please stick around and email in your questions, and we'll get back to those questions to you via email. If you have any, if you want some more information on anything we do as a company, you can visit our website and you can learn about the synthetic guides we make that can be used for R&D. Maybe you're interested in a clinical application. The multiplex knockout libraries, again, those are available for the whole human genome for different protein families. We can make custom libraries. We often make custom libraries here for ourselves. And again, like I mentioned, we're building these gene knockout kits where you can actually take advantage of this optimized approach to knock out a gene very quickly. Or if you're maybe looking for us to do some CRISPR editing for you, we can actually also make these gene knockouts in these CRISPR engineered cell lines for you. That's also a service that we provide as a company. So hopefully you've learned a little bit about how the thinking and the science that went behind how we built this library and how we adopted these methods from utilizing them to knock out genes in our cell lines that we were generating into this library format that you can use for doing any kind of functional genomic screening or assay. So thank you for your time. I appreciate it. And I'm happy to take any questions that you send along. Thank you so much.